Northern New Mexico College is a region-serving institution found here in the Southwest. Like many small colleges, we have limited access to scientific instrumentation. Nevertheless, our passion for science and our capacity to learn is unhindered. In the past, in-depth neuroscience lab experiments were mostly practiced at research institutions with appreciable funding. Our only chances for these experiences were to get accepted to their summer programs, if indeed they were available. However, with the advent of open-source products and software like Arduino, the Spiker Box by Backyard Brains, and Audacity and Python, the idea of neuroscience for everyone can be realized. Our neuroscience lab is developing a low-cost approach solution to bridge the gaps that have been difficult to cross for the average small college. We have repurposed typical lab items like these glass pipettes and pulled them into electrode holders. We have taken disposable micropipette containers and renamed them as water baths. We find outdated computer equipment and salvage what we can. Raw materials from hardware stores are blank canvases. Locally caught invertebrates are perfect model organisms that are low cost, readily available, easy to keep, and they fall below IACUC restrictions. These earthworms were bought from our local live bait and tackle store. We also have colonies of cockroaches, and we house crayfish from our nearby lake. Like any college, we have an inventory of hardware and equipment, sometimes shared across departments, but our goal is to someday share our techniques and spread the optimism. Therefore, the option to run experiments with the least expensive hardware is attainable. Any lab following our model can tailor their configurations based on their available equipment. We utilize our better graded hardware to corroborate any data produced by the low-cost approach. We also run our experiments with mixed configurations to further develop our own research stories. Our research is focused on the curious escape behavior of earthworms. Dr. Ken Cadenia from Vanderbilt University published an extensive study elucidating the predator-prey interactions involved in worm grunting, a localized technique for harvesting live bait. When these worms sense distinct seismic vibrations, they quickly surface. This behavior is attributed to an escape response elicited by the local mole. As mentioned in his paper, the results raised a number of questions from the perspective of neuroethology. Upon further research, we found no literature addressing this specific earthworm escape behavior under the terms of neuroscience. There is, however, a great body of literature involving the earthworm's giant fibers, which mediate the rapid withdrawal escape response. In fact, much of the original work was done by Dr. Theodore Holmes Bullock, one of the founding fathers of neuroethology. Much has been learned about the electrophysiology of neurons and neural networks from such giant fiber systems, but it is often difficult to expand these physiological investigations to a natural setting. Diplocardia might provide such an opportunity. To begin our neuroethology work, we built an apparatus that subjects an earthworm through different controlled frequencies. This is to simulate seismic activity that surrounds an earthworm when a burrowing predator is in proximity. This speaker vibrates a platform coupled by a connecting rod, which is a cork and dissecting pin. The platform floats in a water bath to lower the coefficient of friction, therefore lowering any variables that might attenuate and alter the stimulating signal before reaching the organism. We vibrated a petri dish marked with the quadrants and recorded time spent in each quadrant. We did this with multiple frequencies and noted change in movement per frequency. We then set up a preparation to record extracellular activity from the worm's circumpharyngeal connective. Our preliminary data found an increase in activity within the 25 to 45 Hz frequency range. This raw recording indicated our proof of concept. The green train is a recorded amplitude and frequency that was generated by LabChart software and sent to the vibrating platform. The red waveforms are the simultaneous neural responses. At 200 Hz, we see little to no response. In contrast, the 35 Hz vibrations elicited a burst of neural activity. It is a clear response to stimulation at discrete frequencies. This is a sustained stimulation of various frequencies that are behaviorally relevant, and this is where neuroethology comes in. These experiments have been with the Canadian nightcrawler Lumbricus terrestris, and they are not the same species that Dr. Cadenia studied, the Diplocardia mississippiensis. Nevertheless, these earthworms display an apparent sensitivity to specific frequencies. This raises interesting questions about the earthworm nature and the neuroethology behind their remarkable behavior. Are there special cells tuned to certain frequencies, similar to those in the ear? What is the mechanism mediating these responses? Is the D. mississippiensis surfacing response an inherent trait? Can different earthworm species learn this behavior? Northern New Mexico College may be a small institution, but our ideas are big. We hope to inspire students and educators with similar adversities to join the open source community, and they too can enjoy neuroscience and STEM fields with the resources they have and no burden of confinement. Our lab experiments spark the interest of young scholars by giving them hands-on training with local model organisms, while connecting theory and neuroanatomy. Meanwhile, they are developing neuroscience vernacular and the scientific method and building creative, resourceful, and inventive traits that will carry on through their academic career.